what is a sarcomere? A sarcomere is a carefully arranged bundle of filaments. Now, the reason for the careful arrangement is the different filaments interact with each other. And it's that interaction between protein filaments that actually causes contraction. So, bundles of bundles of bundles. So, a sarcomere, which is this structure, hugely microscopic. Okay, we're down at the EM level. So, the sarcomere exists inside of a cell and is a part of myofibril. Okay, so that sarcomere, and please remember it has a three-dimensional shape. So often when we see the sarcomere, it's presented like this, kind of flat. But remember, if we were to look at this from the side, we would see that it is in fact a three-dimensional structure. It isn't a flat structure, okay? And the sarcomere, which I think I have another slide for you. The sarcomere is made up of thick filaments and thin filaments. And the arrangement of those thick filaments and thin filaments creates a kind of anatomy. So you can see here where it says Z disc, A band, I band. Well, that's what we're getting into here. So if we take and we look just at this ultra microscopic structure, all right, in the light lines here, those are the thin filaments. The heavy, darker filaments that have perpendicular lines in them, those are the thick filaments. The thick filaments and the thin filaments are going to interact to cause contraction. Now, if we look at how things are overlapped, we have a couple of different defined anatomic areas in a sarcomere. Okay, so the A band is the part of the sarcomere that is made up of thick filaments only, right? The Z band, or sorry, the Z disc is where the thin filaments are bound together, right? So we have, and the Z disc is neither actin or myosin. The Z disc is a different set of proteins, which we don't get into and name in this class. But they sort of stabilize the thin filaments in the center. The I bands, that's here, okay, the I bands are only actin. So essentially the I bands are between the A bands. Now, why these funny names? When you look at this under an electron microscope, you can see these areas. Like the A bands are almost dark black. The I bands are significantly lighter, right? So we can see the distinction. So A bands, I bands, Z disc. The Z disc then is in the center of the I band. The Z disc binds the thin filaments together. The A band is made up of your thick filaments. All right, so why this structure? This is one of my favorite pictures of the sarcomere because it has all the important bits in one picture, right? So here's our Z disc aligning and stabilizing everything. Here are our thin filaments, right, reaching out. The thin filaments have troponin and tropomyosin, right? Troponin are the little black dots. Tropomyosin are the little purple spheres on top. We'll talk more about that in a minute. Then we have this molecule called titin, T-I-T-I-N. It's called that because it's one of the largest known proteins in the body molecularly. It's called titin because it has like a huge, huge number of amino acids. Titin kind of behaves like a weak little spring, a spring this way, right? Like a ballpoint pen spring. It's pushing out a little bit. One of the things we'll talk about in a minute is that a muscle can only contract under force. It can't lengthen. Like we don't have a system to make a thing longer, right? Muscles can only shorten. It's why they work in pairs, right? While one is shortening, the other is lengthening passively. Titin, a little bit, pushes a muscle longer, okay? So it's the only part of this whole system that does that. And really, the purpose of titin is to keep the M line the thick filaments and the thin filaments from getting all bunched together, right? So Titan works like a little spring. Now, when this system is activated, the action comes when the thick filaments with their myosin heads interact with the thin filaments and cause muscle contraction, which we're coming up to. But you can't get that until you have the anatomy. So this has, so now that we're getting into the real phys stuff, as you're studying for my exams, be sure that each picture, you know what it is and that you know about it, unless I've told you otherwise, okay? I kind of build my PowerPoints around the pictures, and then obviously I do a lot of talking, okay? So it means that I think of the pictures as kind of the anchor of each slide, right? So be sure that you understand the pictures and the graphs. With every graph you see in my class, I will do my best, and if I don't, correct me, to explain exactly what the axes are and exactly what you're supposed to see in it, okay? Because I know graphs are not everyone's friends, but it does help us to look at the relationships between things. All right, so we have our beautiful sarcomere arrangement. Now let's put this thing into motion. So we're going to zoom in even more. So thin filament, thick filament, right? So that means the Z disc is out here somewhere, and we got a bunch more of these systems, but we're going to just look at one. All right, the thick filament, the heavy chains, that we also call them, are made up of myosin. Myosin has a shape like a golf club, right? It has a long sort of shaft, and then it has a head that sticks up. So long shaft, head that sticks up. If we look at the molecular structure of this, we see that it's almost like somebody's taken a whole bunch of golf clubs and bound them together by their handles, right? By the shafts. So that's kind of what a heavy chain looks like. The thin filaments, the actin chains, have a couple of key components. Troponin and tropomyosin. I think I misspoke on the previous back here. Okay, let me correct. Tropomyosin, long threads. The black things are the active binding sites. Sorry, the myosin binding sites on actin. And the purple ones are the troponin complex. It's easier for me when we put it into motion. Okay, so these myosin heads are going to interact with the active sites on the tropomyosin filament, okay? And that's going to cause contraction. Okay, so in a normal resting muscle, the active sites are all hidden. This is important because if they weren't hidden, muscles would be contracting all the time. And we obviously don't want that. So the active sites on myosin are, the active sites on the thin filaments are covered most of the time. When calcium is present, it causes troponin and tropomyosin to rearrange, exposing those active sites. Once those active sites are exposed, then the myosin heads can bind to the active sites and we can start getting contraction. Now, these active sites are only exposed when calcium binds to troponin. That causes tropomyosin to change its orientation, exposing the binding sites. Now, once myosin attaches to these binding sites, then we get an ATP using process, which causes the head, which typically sits like this, to pull forward. It's like a ratchet system, right? That pulls the thin filament a little bit towards the thick filament. Do that with hundreds of thousands of myosin heads, and then you start to get your contraction, right?